Hi guys, it's Harry here, and today I want to talk about cortical folding, or in layman's terms, why smooth brain means big pain. Throughout history, the brain has been widely known as a complex organ and has been a topic of great mystery for many thinkers, with its first recorded documentation as far back as the ancient Egyptians. Even to this day, research into the brain and clinical treatments of brain-related issues are regarded as complex and challenging.
I mean, it's not exactly brain surgery, is it?
But one mystery of the brain that gets often overlooked is something that it's possibly most recognisable for: its distinct pattern of folding. The structures of this wrinkled surface can be described in terms of its bumps and folds, with the peaks called gyro, a Latin word coming from the Greek gyros for ring, and troughs called sulci, the Latin word to describe the furrow made by a plough. Despite initially seeming insignificant, the reason and mechanism behind the specific pattern of folding has puzzled neuroscientists for years. Firstly, why does the brain fold in the first place? Is it just to increase its surface area? Well, although cortical folding does increase the surface area of the brain, this does not explain the full story behind it. Cortical folding occurs prenatally, so before birth, and although there are some size constraints put on the developing brain by the skull, this is not the main reason for cortical folding, because the cranial bones do not fuse until around 12 to 18 months after birth, meaning that the cavity within the skull increases as the brain grows postnatally. Also, there must be something controlling how the brain folds, because if it were truly random, then we wouldn't see the same folding patterns that are not only shared between individual humans, but also between species. So what is this thing that controls cortical folding? To answer this question, let's look at what makes the brain fold in the first place. Later described as the Van Essen hypothesis, David Van Essen proposed a tension-based theory in 1997, suggesting that neurons pull the cortex inward at the base of the soon-to-be sulci. However, this theory was later refuted since the tensile forces in the neurons were displayed not to be in the orientation required to achieve this. The more widely accepted hypothesis actually comes from a 1975 paper by Dr. David P. Richman and his colleagues, which has more recently been supported by more sophisticated mathematical modelling and experimental testing. This hypothesis, known as the differential growth hypothesis, describes that if two layers attached to each other expand at different rates, these resultant forces will result in buckling. More specifically, if the outermost layer grows faster than the layer below it, it will result in the recognisable bumps and grooves of gyri and sulci seen in the brain. The pattern of folding is not only determined by the relative distance between the expanded layers, but also by the thickness of these layers, and the difference in the relative elasticity of the two layers. Okay, so the upper layer grows at a faster rate than the lower layers, but what causes this? This comes down to a couple of cellular processes, both relating to the migration of immature neurons. The first is neurogenesis, which is the production of neurons from neural stem cells. These immature neurons travel along the radial glial cells to the outer surface of the cortical plate, the embryonic precursor to the cortex. This process is known as radial migration. It has been shown that in folded or gyroencephalic brains, that the regions undergoing greater surface area expansion, and thus greater folding, exhibit far greater levels of neurogenesis than those with little cortical folding. Besides the radial migration associated with neurogenesis, gyroencephalic brains also display another type of migration, tangential migration, where rather than the developing neurons moving towards the outer surface of the cortical plate, they move sideways within the layer itself. This results in differing areas of neuronal density in the cortex that is not seen in lissocephalic or smooth brains. Since cellular composition and density plays an important role in the structural properties of the cortical plate, such as elasticity and size, this makes sense that the genetic expression and associated cellular signaling pathways play key roles in governing cortical folding. The sheer number and complexity of the genetic and molecular factors involved in the cortical folding would be too long and too mind-bogglingly boring for this video, but as a good rule of thumb, a vast majority of the genetic and molecular interactions in the process of cortical folding are involved in the regulation of neurogenesis and the neuromigration. In fact, the most prominent folds have been demonstrated to show genetic banding patterns that are not seen in lissencephalic animals. So now we know a bit more on the mechanistic, cellular and genetic mechanisms underlying cortical folding, we can now better appreciate the development of cortical foldings in humans. During brain development in embryology, the pattern of gyrification only appears after all cerebral cortex cells are created and positioned in their proper location and layer. In humans, this process consequently takes place in multiple phases, referred to as primary, secondary and tertiary folding. Beginning at gestational week 27, in primary cortical folding, gyrification patterns appear that are most similar between individuals at consistent location and inconsistent size. In the second and tertiary phase, occurring at week 31 and week 37, respectively, the gyrification shows more randomness and variation between individuals. This entails that just like your fingerprint, the pattern of cortical folding is, despite general similarities, unique in each individual. Interestingly, not in all species, these latter two phases occur. Typically, only larger species, such as humans, whales and dolphins, show secondary and tertiary gyrification, and smaller species, such as cats and ferrets, only undergo primary gyrification. After these three phases, gyrification is complete. This means that after birth, the pattern of cortical folding no longer develops. When cortical folding goes wrong, the patient can suffer severe side effects. Symptoms start usually only a few months after birth, usually presented as developmental delays. In a case study, researchers describe a 5-month-old patient who presented to A&E after the parents were worried about abnormal movement. The patient showed more episodes of flickering of the eyes and tonic movement. He had stopped fixing and following, stopped responding to loud noises, stopped reaching for objects, and he'd stopped babbling, things he always did before. Development had halted at 4 months of age. The posture of the patient was frog-like and very floppy. Blood testing revealed nothing out of the ordinary, but an EEG indicated hyperarrhythmia and MRI was performed. On this MRI, it is very clear to see that the patient has few sulci and a thick cortex. All signs pointed to type 1 lissencephaly. Genetic testing showed a mutation in the LRS1 gene, confirming the hypothesis. Prognosis is relatively poor, with most patients passing away at the age of 10. Lissencephaly causes severe developmental problems with milestones not being met and seizures. So having no folding is bad, but actually having more folding is also not particularly good either. A disease where patients have excessive microfolds is called polymicrogyria. Compared with lissencephaly, the symptoms have some overlap, mainly developmental delays and seizures. Because most of the time only a specific part of the brain is affected, the symptoms correspond with the brain area affected. For a case study of 12-year-old male with homozygous ADGRG1 nonsense polymicrogyria, developmental delays were observed. From the timeline of symptoms, a similar pattern could be observed as the lissencephalous patient. Symptoms started a few months of age, milestones are not being met, motor control is underdeveloped, seizures start to emerge, and cognitive impairments. The patient slowly progressed, and at 21 months and about 2 years, was able to maintain cephalic control and an unaided sitting position. He was able to maintain eye contact, smile, and produce elementary sounds, but language was not present. However, at 2.5 years, he worsened and suffered significant motor and cognitive regression. He also lost cephalic control and wasn't able to sit upright unsupported. At the same time, seizures worsened with multiple types. Now you might ask, does folding occur in all species? The answer is no. Birds, reptiles, amphibians and fishes do not show cortical folding. Due to evolution, they have enlarged specialized regions in their brain, like the hypostriatum and vulst in birds. But even some mammals have a lissencephalic brain. Studies show that species with cortical surface below 400 meters squared are lissencephalic. For example, rats. So if not rats, what would be an ideal model animal for studying cortical folding? Scientists use the ferrets because they resemble human brain structures, have short gestational period, but most importantly, cortical folding occurs postnatally. Where did these species separate from the folded species? Who is our common ancestor? As gyrencephaly also occurs in some non-placental mammals, including echidnas and kangaroos, it seems likely that this trait arose in the ancestor of all mammals. The gyrification index, or GI, which is defined as the ratio of total neocortical surface area divided by the superficially exposed neocortical surface area, shows a strong positive relation with brain mass. A GI of 1 indicates a completely smooth brain, and GI greater than 1 indicates increasing levels of cortical folding. Humans, with a GI of about 2.56, rank highest among primates, but are less gyrencephalic than some other animals of other orders, such as zebras, with 2.94, and elephants, with 3.81. However, the relationship between the GI and brain mass is not strict. Some species, such as beavers, manatees, and humans, have a smaller than expected gyrification index. Going by brain mass, humans have an unusually thick neocortex of around 3.4 millimeters average, which is exceeded only by the manatee cortex thickness of around 4.0 millimeters. A recent study shows the protein TRNP1 co-evolved with cortical folding in mammals. So what is this protein and why is it important? TMF-regulated nuclear protein 1, i.e. TRNP1, plays an important role in cortical folding. It shows that TRNP1 does not significantly correlate with brain size, but it does significantly correlate with the gyrification index. That rate of protein evolution of TRNP1 correlates with the gyrification in mammals, and that the activity of regulatory element of TRNP1 co-evolves with the gyrification in catarrhines. Overall, the process of cortical folding seems quite simple, but the mechanisms of regulation are vast and complex. This large system of regulation involves an interplay between the mechanical molecular processes and genetic expression. Obviously, we have left out the specifics of a lot of data, but if you are interested, here are some resources for further reading. For now though, make sure you like, subscribe, fuck it, I am never doing this again. This has broken me. Please stop.